Okay, hi and welcome to this lecture. So this is the third lecture in the Bioinformatics Resources and Databases section. So hopefully you will have a good idea now about the various databases, bioinformatics databases, and how to search them. So in this lecture we're going to focus on DNA sequence analysis. So this is very basic sequence analysis. So it's really about finding features on a DNA sequence. So we need to understand how to extract a DNA sequence from a database, which you will hopefully have done already, and then use online tools to be able to do simple manipulation to find features on the sequence. So the learning outcomes is to find these features, be able to save them to get DNA sequences in the right format, look for features like restriction enzyme sites, any features necessary for cloning or for PICR, and then we want to be able to interpret the analysis results and understand the biological impact of functional regions. So there's two major components to bioinformatics. There's really storing and retrieving of data, which you've heard about, and then there's the manipulation of data. So once you've got data, there's something you want to do with it to answer a particular research question. So what I'm going to focus on is finding features on sequences. There'll be other modules that will cover sequence similarity searches and the protein side of things. So I'm looking really here at DNA. So if you think about a strand of DNA, the DNA obviously encodes all the information necessary for life. So upstream of a potential coding region, you have promoter regions, you have regulatory regions. Not all pieces of DNA actually encode a protein, so there are intergenic regions that have other functions as well. Then from the protein coding section, you will then have the process of transcription will get the DNA to mRNA, and then translation gets that to a protein. So what we want to do in this module is find these features. So it's basically positions on a DNA sequence that have a particular function. So we're going to look at translation, getting from the DNA to the protein. We'll look at various aspects of cloning and restriction enzyme analysis and PICR. And then I'll just briefly mention some of the analysis you could do with RNA, but there won't be a focus on that. So we can assume that a sequence is retrieved from a database, and that's because you'll have done it before. So here we'll focus on a few things like how would you convert to different formats, because there are different formats for saving sequences, a note on accession numbers, because every time you go to a different database there'll be a different accession number for the same sequence. And then, as I mentioned, restriction enzyme analysis, primer design, finding features, and I'll just touch on gene prediction. So, and then RNA sequence analysis. So for sequence formats, the main format that you'll find a DNA sequence in is Fasta format. And the format is simply a line starting with a greater than sign that describes the title of the sequence. And because the greater than sign is at the beginning of the line, many programs know that that is just the name of the sequence and not the actual sequence. And then that's followed immediately on the next line by the sequence itself. If you have multiple sequences in Fasta format, they immediately follow the next one after the end of the previous one on a new line. So this is a well-recognized format. There are other formats, however. So there's GenBank format, where there's a bit of information about the particular sequence. Usually it comes from GenBank, so it needs this annotation. And then these lines are actually numbered, so it's just a different format. And you can usually convert between sequence formats in most sequence analysis programs by just saving it in these different formats. So some of the things you could do with the sequence overall is look for the DNA composition. So this would be the percentage GC content versus AT content. And this is important because, so you have G bonding to C and A bonding to T. This is important because GC bonds are stronger than AT bonds. So, for example, when you're doing primer design, whether the percentage of GC versus AT will determine the annealing temperature. So it's important to have two primers match in terms of their GC content. Other reasons you might want to look at GC content is that if you want to identify regions in the genome of horizontal gene transfer, there often this a difference in percentage GC content in a particular region that's different to the overall GC content of the genome might be some indication of an insertion from another genome. And then gene prediction, because there's also a slightly different GC content in coding versus non-coding regions. So just a note on accession numbers. Every database you go to will have their own accession number system, and it doesn't matter as long as you know how to get from one to the other. So here's some examples of a GenBank versus GenPabt versus UniProt. You don't need to learn what they look like, although you soon will become familiar with them. But the main issue is how you get from one to the other. So you might have exactly the same sequence in Ensemble, AMBL, HGNC, etc., and they just have different accession numbers. So many people behind the scenes who work on these databases have worked closely together to try and map between accession numbers, to say if this accession number in AMBL has exactly the same sequence as this accession number in Ensemble, then those two can be mapped to each other. And there are a couple of tools which can allow you to switch from one to the other. So one example is the UniProt website mapper. And then there's PICR, which is another tool, protein identification cross-reference resource. So what these do is you'll input one or more accession numbers of the ones that you want, and then you tick off the databases where you want to find the corresponding accession number. And then it does the conversion. So in this case, the accession number was on the left 060260. This is a UniProt accession number. That's a protein accession number. And it's finding now all the list of proteins in Ensemble that have that same sequence in RefSec and in some of the other UniProt databases. So that's just a few general pointers about sort of overall sequence analysis and accession numbers. Now we'll come down to actually doing some sequence analysis. So as I mentioned, I'll touch briefly on restriction enzyme analysis, primer design, and finding features. So if you want to do some cloning in the lab, what you need to do first is say what piece of DNA do you want to clone, so and what is the sequence of that DNA. Then you will need to identify restriction enzyme sites in these and match these to restriction enzyme sites in a cloning vector. So for those who are not familiar with cloning or restriction enzymes, so restriction enzymes are made naturally by various different bacteria, and they cut specific sequences, recognition sequences in a DNA sequence. So the recognition sequence varies for different enzymes. So some are 4 base pair recognition sequences, some are 8 base pair recognition sequences. The other thing to note is that you get different products. So you get what's called blunt end cutters. So in this example with high 3, you can see that the recognition sequence is GGCC, and then the other one is just the 3' to 5' reverse complement. And in this case, the enzyme cuts straight down the middle, so that when you get to the product, there's no overhang. So that's what's called a blunt end. Then you get sticky ends, which means there is an overhang. So in the second example, you'll see that the enzyme cuts on the top strand between the G and C on the 3' side, and the bottom end between the G and C, again on the 3' side. This leads now to an overhang of the CG, and then on the other strand, the overhang of the CG. And then you get, that would be a 3' overhang, and then you get a 5' overhang, which is just the opposite. So the opposite, the 5' end is what is the sticking out compared to the double-stranded section. So everything where the dotted lines are here would continue to be double-stranded DNA. So there are various different tools that will predict these. Because they're known binding sites, the tools are basically just pattern matching to find those particular sets of patterns in a DNA sequence, and the output would be, for your double-stranded DNA, the list of potential restriction enzyme sites. So when you're cloning, what you want to do is you have your vector. Now, a vector usually has some features on it. So it has a multiple cloning site that interrupts a gene, in this case the LAC-Z gene, because this is a way to identify bacteria which have had this plasmid in it and have had the gene interrupted. So in this particular case it turns blue or white. I'm not going to go into the biological details. Then you also have a drug resistance gene, so that if you grow the bacteria in the presence of this particular drug, so an ampicillin, then only bacteria that have taken up this plasmid will actually be able to grow. The rest won't even bother to grow. So you'll be able to see, did it grow and what color is it, yes or no, did the insert go in based on the color. So what we're doing in cloning is we have a list of enzymes that are available in the multiple cloning site for the vector, and then we know what the restriction enzyme site is for our DNA. So we find some appropriate enzymes in the right place that cut in the right place. You cut your DNA, your double-stranded DNA with those, and now you have the overhangs, so hopefully preferably different enzymes on each side, and then you find those overhangs when you cut the vector, and the vector then opens up, finds the sticky ends that are at the ends of these, which now match the sticky ends of those, and the red sticky end here will stick onto that, and the red-green sticky end on this side will stick onto this side, and your insert gets inserted into the vector. So once you've done cloning, you might then want to do a sequencing experiment just to make sure your clone, the insert is correct. So you sequence that little piece. What you would need to do though is potentially remove vector sequence, because sequencing primers tend to be in the vector sequence. So if you want to just see the sequence of your insert, you need to remove these vectors. And so you can do this just by running a blast search against the UniVec, which is basically a database of vector sequences. So it's very simple. And obviously you hope that the vector is only at the ends and not somewhere in the middle of your sequence. So that's really cloning and how you would use restriction enzymes. So you would use a program to identify potential restriction enzyme sites. You map those then to what restriction enzyme sites are in your cloning vector, and then you use those to determine which enzymes to cut with. And also which enzymes you choose will also help you to determine which way the insert will be inserted into the vector. So you can drive it. That's why you would cut with two different enzymes, so that you can really drive it in a particular direction. So then moving on now to primer design for PCR. So polymerase chain reaction is basically an amplification process. And the idea is this. So you have double-stranded DNA. Then you design primers that match to, so they would bind to, they're complementary to a particular region on one strand where you want the insert to start. And then a second primer in the reverse complement on the other strand, on the other end of the piece of DNA that you want to amplify. Then this goes through a process of multiple different temperatures. So you throw these primers in. You firstly heat the DNA to 95 degrees so that the DNA strands separate. Then you cool it to a temperature that's appropriate for the sequences of these to bind to each other. That's called the annealing temperature. And then you have an extension period. So this is, we use Taq polymerase, which can work at high temperatures. And this now then continues as though you're doing DNA replication and just synthesizes the strand in that direction, and the synthesizes the strand in that direction. And then you end up now with two double-stranded copies of just this piece that you wanted to amplify. And then you go through multiple rounds. And then you obviously get an exponential increase in the number of just this insert, number of copies of just this insert. So when you're doing primer design, the primers should be similar in length. And they should be similar in annealing temperature, because you want to have the optimal single annealing temperature that both of them bind. And you also have to make sure that the primers only bind once in the genome, and that is to the exact place that you want to start or end your insert. If they bind in more than one different place, you're going to get multiple different products, and that's going to be difficult to separate the one that you want from the ones that are that you don't want.